Bismillahirrahmanirrahim. Assalamu alaikum dear students. This is Dr. Azam Ali from Department of Biochemistry, FRPMC. The practical that we are going to discuss today is about the phenomenon of surface tension. Learning objectives are to understand the phenomenon of surface tension, to understand the biomedical importance of surface tension in human body, and to demonstrate the phenomenon of surface tension by Hayes sulfur test. So what is surface tension? It is defined as force in dynes acting upon a line 1 centimeter long on the surface of liquid. In a liquid, there are intramolecular forces that are termed as cohesive forces. In the interior of the liquid, these molecules experience cohesive forces from all the directions and they can move in all the directions. In contrast, molecules at the surface of the liquid do not experience cohesive forces from above due to air-liquid interface. As a result, intramolecular forces or cohesive forces along the surface are strong. They tend to contract the molecules and form a thin film at the surface. So the force or tension required to break this film is known as surface tension. We also come across surface tension in our daily life as well. For example, surface tension is responsible for a spherical shape of the liquid droplet or the formation of lower and upper meniscus of liquids when it is taken into a cylindrical container. So due to strong cohesive forces, these molecules tend to pull the liquid droplet into a spherical shape. Also, beside the cohesive forces, there are adhesive forces as well. And these are attractive forces between the liquid molecules and the container. So in the formation of lower meniscus, there are strong adhesive forces than the cohesive forces, and in this instance this is mercury, so it forms upper meniscus because there are more cohesive forces and less adhesive forces. So many agents tend to lower or enhance the surface tension. For example, soap, detergent, bile salts and proteins tend to reduce the surface tension. And salts such as sodium chloride, they increase the surface tension. The unit of surface tension is either dynes per centimeter or ergs per centimeter square. So the surface tension of water is 72 dynes per centimeter at 25 degrees Celsius, and that of normal urine is 66 dynes per centimeter at 25 degrees Celsius. Biomedical importance. In our body, the phenomenon of surface tension affect different physiological processes. So there are counter mechanisms to prevent those effects. For example, bile salts facilitate in the emulsification of lipids. These bile salts are amphipathic steroidal biosurfactants that are synthesized in the liver from the cholesterol. They are synthesized from conjugation of bile acids with either taurine or glycine. These are the two amino acids. And these bile salts have amphipathic nature, which means they have hydrophobic sides and hydrophilic side as well. So they break down large fat globules in our gastrointestinal tract into small droplets. These are the emulsified droplets, and thus helping in the absorption of lipids. So another example of biosurfactant is the pulmonary surfactant lining the alveoli, and this surfactant lower the surface tension in the thin alveolar fluid film. So as a result, there is reduced work of breathing. This biosurfactant is synthesized by the type 2 pneumocytes in the lungs, and it's composed of phospholipids which include lecithin and sphingomyelin at the ratio of 2:1. Although the synthesis of surfactant starts in the intrauterine life by 28th week of gestation, however, in the case of preterm birth, the ratio between lecithin and sphingomyelin is disturbed. As a result, there is a condition known as respiratory distress syndrome appears in the preterm babies. So the normal urine surface tension is 66 dyne per centimeter. So in case of abnormal presence of bile salts in the urine, the surface tension is reduced to 55 dynes per centimeter. So as discussed previously, bile salts are amphipathic in nature, so they tend to lower the surface tension of the urine, and this is utilized in the Hayes surface test, in which sulfur powder is sprinkled on the urine surface. So in case if the urine is normal, the sulfur powder will remain on the surface, but if there is presence of bile salts, the surface tension will be lowered and the sulfur powder will sink towards the bottom of the test tube. So in this practical, we will perform Hayes test on different substances and see if they increase or decrease the surface tension. So the reagents required for this practical are distilled water, 10% NaCl solution, a diluted bile salt solution, a soap solution, and a detergent solution. And we also require a finely powdered sulfur powder. So to perform this practical, we will take 5 test tubes and label them alphabetically or with the names, and fill these test tubes to half of the capacity with respective reagents. Then we will sprinkle finely powdered sulfur on the surface of liquid in each test tube. Sulfur powder will remain on the surface of test tube which contain water and sodium chloride solution because of high surface tension. And in case of liquids containing bile salt, soap, and detergent, the sulfur powder will sink towards the bottom. Okay, students, we will now perform a Hayes test to demonstrate the effect of different agents on the surface tension. For this, we have taken 5 test tubes and labeled them with the names. The first test tube contains distilled water, the second one contains 10% sodium chloride solution, the third contains bile salt solution, and the fourth one contains soap solution, and fifth is made up of detergent solution. And we also have finely powdered sulfur powder as well. Okay, students, we will now add sulfur powder to these test tubes one by one. First of all, in the distilled water, and in this you can see the sulfur powder is still afloat and it has not settled down in the bottom because of the surface tension. Now I'll add the powder to the second test tube, which contains 10% sodium chloride solution, and still the powder is still afloat. And now we'll add the powder in the third test tube, the test tube containing bile salt solution. Okay, we'll now add powder to the fourth test tube that contains soap solution, and again the powder has settled down in the bottom, which shows that the surface tension of this solution was also lower than the sodium chloride and water. Okay, we'll add sulfur powder to the fifth test tube, which contain a detergent solution. So similar to the previous two solutions, it also has a lower surface tension. That is why the powder has settled down in the bottom.